are returning to the river as adults, as mature adults. At that point, we'll artificially spawn them. So this is a female getting her egg stripped via an air pressure system. At that point, we'll separate her eggs into different clutches and either fertilize it with one male sperm or multiple male sperm. At that point, the embryo is created. It gets placed in one of these heat tray incubator stacks for a few months. At that point, they'll develop and hatch. And then this is the point, once they've used all of their egg reserves, they begin exogenously feeding. So before we move them outside, we feed them indoors to keep the environment a little more controlled so that we're not going to have major losses once we move them outside. So after a couple of weeks, we do move them outside and we rear them to about this size, which is between 120 and 200 millimeters. And this is what we refer to as a smolt. And this is a fish that's still residing in freshwater, but if it was released into the river from the hatchery, it would begin an out-migration towards the ocean. And so I'm going to be talking about smolts from now on, and that's what I'm referring to, a fish that would be released from the hatchery and is about to go into the ocean. So we release these hatchery fish, they go out to the ocean, they feed, a lot of them die unfortunately, but we do have hatchery fish that return. And when they return, they're looking to complete their life cycle like any other fish. So we have these hatchery fish returning to the river. They do in fact spawn, so this lighter gravel area is indication that salmon have spawned there. And they do in fact produce viable embryos that hatch into little baby fish that then grow up and turn into smolts. So you may be asking yourself at this point, what's the issue, right? We bring fish in, we rear them up, there's really high survival, we let them go, they come back, they spawn, and they produce smolts in the next generation that then go out, come back, so on and so forth. Well, a little bit of a Q&A here. So we have two columns of fish, the fish on the left and the fish on the right. Can anybody tell me the difference between the two? The ones on the left don't have an adipose fin, which means all of these fish are hatchery fish. And the other big thing that has come out of Mike's lab and other labs throughout the Pacific Northwest is these fish, because of their hatchery origin, when they return and spawn, return half as many offspring as the fish on the right. And this was borne out in a recent review paper done by Mark Christie. So RRS is a measure of the number of offspring returning per fish. And so I'm going to be using this RRS and be talking about it throughout the rest of my talk. And so it's a measure of fitness or a measure of performance. And this 0.534 number is at 50%. So we know there's fitness loss being caused by hatchery rearing, but there are two potential drivers of fitness loss in these hatchery fish. The first could be a heritable genetic effect, so changes in the gene or selection for a specific phenotype in the hatchery environment. The second could just be a main effect of the environment a fish was reared in. So hatchery fish, from egg stage to that smolt stage, are reared in a relatively benign environment. No predators, relatively little disease, ample food, and lots of buddies to swim around with. In the wild environment, a fish has to hatch, defend a little space, grow, avoid predators, avoid disease, a whole slew of pressures that you don't necessarily see in the hatchery environment. So the difference could be potentially due to environment. However, there is evidence showing that the fitness loss is caused by genetic effects, and especially in steelhead. And what I'm going to present is two pieces of evidence in steelhead that show there is a genetic effect of fitness loss, because that's really what my dissertation is focused on. The first piece of evidence showing that fitness loss is genetic is we can look at that relative reproductive success of wild-born adults with different hatchery ancestries. So here we have a yellowfish, which is a hatchery fish with no adipose fin. So it was released from the hatchery, went out to the ocean, and is going to come back and spawn. This is a wild fish that is coming back to spawn, never saw life in the hatchery, never saw the hatchery at all. So these two fish spawn in the river, and they give rise to this generation of fish. So we have what we would term a wild fish because it still has its adipose fin intact, but it had two hatchery parents, whereas this wild fish had two wild parents. Now what we can do is assess the relative reproductive success or the number of offspring that these two fish return in the next generation to see if there's a genetic effect. And this was done in the Hood River by Hitoshi Araki and Mike. And what they found was this wild fish with two hatchery parents returned 30 to 40% of the offspring of this wild fish with 2 wild parents. So the important part here is there was no difference in environment. This wild fish with 2 hatchery parents and this wild fish with 2 wild parents experienced the same environment. Neither of them experienced the hatchery environment at all. So the only difference here is really the genetic lineage from which they came. So this is strong evidence that there is a genetic effect to fitness loss. The second piece is looking at a performance trade-off. And this was work done by Mark Christie in 2012 and also coming out of the Hood River. So on the x-axis here we have the number of returning offspring per parent, and on the y-axis we have per capita offspring success in the wild. It's a mouthful even for me. So what I'm going to do is I'm going to talk you through the story of that point and that point there. And this will hopefully clear things up. So what this number 70 means is there was a parent, or there was a salmon that was brought into the hatchery, it was spawned, and it returned 70 offspring as adults. When those 70 offspring, on average, when those fish went and spawned in the wild, so the next generation, they returned less than 1 fish per adult. So they have really high, this lineage, genetic lineage, has really high success in the hatchery and really poor success in the river environment. Contrasting that, we have this fish who returned, this hatchery fish who returned 1 or 2 fish as offspring. And when those offspring went and spawned in the wild, they returned, between 5 and 20 offspring. So there's this really large performance trade-off. Either you have traits that make you really fit in a hatchery and not in the wild, or you have traits that make you a relatively quote-unquote poor hatchery fish and return relatively few, say between 0 and 10, but then in the wild environment you return very many more offspring in the next generation. Now, the other important part about this graph is there's no real 50-50 trade-off here. Because if there was a 50-50 trade-off, all the points would be on a linear line from these points down to that down to that 70. So there's a really big trade-off in that you're either good in the hatchery or in the wild. There's no happy medium here. And so this is another piece of strong evidence that there is a genetic effect to fitness loss. Now there are 2 ways you can begin to attack this question of what's driving fitness loss in the hatchery. The first is you can look at specific traits that could be under selection. The second is looking at the environment that is driving the ability for selection to act. And so I took a 2-pronged approach and I addressed both of these questions in my dissertation. For the first question, I explored if growth rate was under selection in the hatchery. And in the second question, what environments, I explored if high rearing density was driving the ability for selection to act. So right now what I'm going to do is dive into this growth rate chapter. So this was my first big set of experiments. And I'm going to talk you through a little bit about why we think growth rate was under selection. So in salmon and steelhead, the size and release from the hatchery as a smolt is correlated with the probability of survival or the probability of return. And for steelhead particularly, we know the survival curve has this sort of shape. It's a logistic curve. And the important part about this is over a very narrow window of body lengths, there's a rapid increase in the probability of survival that then plateaus out. You don't get much higher after you get above a certain body size. This is going to be important later, but for now, just remember that the bigger you are, the higher probability you're going to survive. The second piece of evidence that shows growth rate could be under strong selection is in the hatchery, we take fish from egg status, or egg stage, to smolt stage in a single year. In the wild, you go from egg to smolt in at least 2 years, if not more. So in the wild, you have a much longer time to achieve the smolt size before you out-migrate. So in the hatchery, if you simply don't grow big enough to reach this stage, you're not going to survive, and you're not going to reach this nice silvery coloration which prepares you to go out to the ocean as a smolt, which is an indicator of how ready you are to go. So at this point, I sought to answer that question. Is growth rate under selection in the Hood River hatchery? And the reason I used the Hood River program was because there's all this strong evidence for genetic effects in the Hood River. So the way I did this was I did it over 2 years, and I'm going to go through each year one by one, because the designs were slightly different. So in the first year, I had 2 different types of crosses. I had 2 hatchery parents paired together, which would produce hatchery families, and then I had 2 wild parents paired together, which would make wild families. Now, one caveat with this design is we can't separate environmental from genetic effects. So an environmental effect here would be something such as differences in egg size. If all the hatchery fish had larger eggs than all the wild fish, and we saw a difference in growth rate, and we saw a difference in body size, we wouldn't necessarily know if the effect was truly due to the genetics in the hatchery individuals, or if it was simply due to larger egg size. So that's something to keep in mind. So what I did was I created 18 hatchery families and 14 wild families. From each one of those families, I put 15 fish into this tank here, and this is where the Hood River fish are reared as a production class, so it's the same facility. And I tried to limit the environment as much as possible by using one tank. There was no replication here, it was one big test of this hypothesis. At that point, I reared them for a full year, or the hatchery managers and staff reared them for a full year, and at the end of that year, I took, for each fish, we measured the length, the body length, and I also took a fin clip for genetic analysis. And to assign fish back to whether they were a hatchery family or a wild family, what I used was genetic parentage assignment. I wish I had time to go into this today. It's a really cool method, and it's actually fairly simple, but I did too much with my PhD, or with my dissertation. I don't want to get through all that, and so I don't want to waste too much time here with this method. But suffice to say, I used 6 microsatellite markers that allowed me to determine which juveniles matched to which parents. And I used the Solomon parentage program to use this, which is a software program by Mark Christie, because I'd still be doing parentage right now if I didn't have a supercomputer. So once I had all the data, to analyze, what I used was a linear mixed effects model. And what a mixed effects model is, it has fixed effects and random effects. Fixed effects are things we care about, we want to estimate. Random effects are simply a factor that we know is going to affect our response, so juvenile length, but we don't necessarily care about. We just want to have it there to soak up some variation. So the first thing I was interested in was, does juvenile length depend on the fish type that you are? Are you a hatchery or wild fish? I also included fixed factors of mother's length and father's length, because it's known if you come from a larger mother or a larger father, you're going to be a larger offspring. I also included a spawn date term, and the reason I did this is not all of those crosses were created on the same day. Some of the crosses that are created earlier on in the spawning season are developing at colder temperatures, because what the hatchery managers do is they chill the water so that all of the fish throughout the course of the spawning season hatch at the same time, begin feeding at the same time, so it makes their life easier, so they don't have multiple batches of fishes at multiple life stages. And then I had some interactions, so I had hatchery or wild versus, I had fish type by mother's length, fish type by father length, so on and so forth. And what this told us was if the effect of mother length was different for the hatchery fish versus the wild fish. I also included a mother length by father length interaction to account for non-additive genetic effects, so gene interactions. And lastly, the mixed part of this model, we know family has a really strong effect on juvenile length, and so we wanted to account for that. And so I had family as my random effect. Now jumping right into results of this year, or for year 1, what we found was fish type had no effect on juvenile length at 1 year of age. We did find a significant effective spawn date, so being spawned 30 days after the first spawn date, so a month later, reduced juvenile length by approximately 1.8 centimeters at that 1 year of age. And I'm going to talk about this a little bit more in a second. You'll notice we also had a fish type by mother length interaction, and that was significant. For wild fish, the effect was the bigger the mom, the bigger the offspring. For hatchery fish, it was the bigger the mom, the smaller the offspring. Now this result was driven entirely by one abnormally large hatchery female. So when that female was removed from the analysis, this result disappears, so I'm not going to spend a whole lot of time on it because this result is truly driven by one female out of a total of 36 female parents we used. So getting back to that spawn date effect. Here on the x-axis we have the date of spawning, and then we have our measure of body length on the y-axis. Each one of these little gray dots is an individual fish. So this cloud of points belongs to 29 April. This dark circle here is the mean fork length, or the mean body size, of this entire cloud of points. Okay? So as you can see, if you respond in late April, early May, you're larger than if you respond in mid-June or even early June. And the reason I think this is occurring is getting back to that chilling effect of the embryos during development. If you chill an embryonic salmon as it's developing, that could reduce its metabolism, which frees up energy for other body functions such as somatic growth. So by using less energy on maintenance or something like that, you actually increase the amount of energy that you can use for somatic growth. And I believe that the early spawn fish were larger at the time of ponding, so when they were moved outside, and I hypothesize that that carried them through all the way through that first year. And what was truly remarkable about this result is differences in size at this stage, when you're a tiny little fry between 30 and 40 millimeters long, translated to 1.8 or nearly 2 centimeters of difference when you were 1 year of age, and approximately 150 to 200 millimeters large, or 200 millimeters big. So that wraps up year 1. Now I'm going to start talking about year 2, and I had a different spawning design in year 2. The reason I did this was my 2x2 matrix here allows me to discern genetic effects from environmental effects. So in the first year I couldn't discern the difference between those two. With this study's design, I would be able to. So what I did was I created these 4 fish types. So I had 2 different types of mom, hatchery and wild, and 2 different types of dad, hatchery and wild. Created 4 different fish types. So we have hatchery, hatchery wild, so on and so forth. The important part with this is this yellow fish and this green fish, because they were mated with the same mother, they're half siblings. Okay? So just like the previous year, I made quite a few of these matrices. I had 16 in total, and 15 fish from each cell, so 60 fish from each matrix, were put into this tank here. Now this was much closer to the production level rearing density than the first year, so it was a more stressful, more crowded environment. In the first year, my fish were reared at a fairly low density. And so this environment is much more like what the Hood River hatchery production steelhead would experience. And again, I had 1 tank, and it was a common garden experiment, so I'm eliminating as much environmental variation as possible. So just like the first year, I reared these fish for a full year, sampled them, took their length, matched their genetic profiles back to the parents, and did a very similar data analysis. However, the one thing that I did have to change was, instead of having fish type as hatchery or wild, because of my spawning design, I needed to break it into mother type and father type. The rest of the model stayed the same. So we have the fixed effects of mom, dad, spawn date, the interactions, and so on and so forth. Now, the other thing that I was able to do with this data set, because I had that 2x2 matrix design, was look at the effect of having an extra hatchery generation on juvenile length at 1 year of age in a pair of half siblings. So basically what I'm going to be doing is comparing this hatchery family to this wild hatchery family, and the only difference between these two is the genetics of the father. They were created from the same egg, the same mother. Okay? And we can do that for wild fish as well, so we have the hatchery mother comparison and the wild mother comparison. Now, if growth rate was under selection in the Hood River program, this is what we would expect to see. So we have father type on the x-axis, hatchery and wild, and then just body size on the y-axis. That triangle there and that triangle there are a pair of half siblings. So this is that yellow family, and this is that green family. Okay? So what we'd expect is the hatchery father to be larger than the wild father and a common mother. And that would go for all of our crosses. So all of our 16 matrices, we should see this triangle being higher than this triangle. So getting into the results, In year 2, just as in year 1, we didn't find an effect of fish type, which was truly surprising, given that we know larger size of release correlates with higher probability of survival. There was an effect of mother length, which is not entirely surprising. That's been known since the 70s or 80s. If you have a bigger mom, you tend to be a bigger kid. Now we look at that paired t-test, so the effect of that extra hatchery generation. And remember, the expectation was this dot was going to be higher than its corresponding half-sibling. Because we see all of these lines sort of crisscrossing and going every which way, we do not have any support for the hypothesis that growth rate was under selection. We didn't see the hatchery fathers outgrowing the wild fathers in a common mother. And if you're the statistics type and you want to see p-values, there you go. Not even close. Okay? So at this point, sort of summing up that first series of experiments looking at if growth rate was under selection, we didn't find evidence for it. Now there are a couple of explanations to explain this. First, perhaps growth rate truly isn't the trait under selection. Maybe it's a trait that's correlated with growth rate, and growth rate just sort of gets swept along for the ride. And so that could be happening in the Hood River program. Alternately, what I found out with this series of experiments is it's incredibly difficult to mimic production level dynamics on an experimental scale. So the amount of stress that the production level fish experience, I'd argue, is probably much higher than the amount of stress that my experimental fish experience. And this is important because differences in fitness between different genetic profiles are much easier to see and much easier to find in stressful environments. So perhaps I just didn't stress my fish enough to create a difference that I would be able to statistically see. So this wraps up the first part of my dissertation, and I'm going to switch gears now and start looking at the environments that might be driving the ability for selection to act. And specifically, I'm going to be talking about rearing density, and the really high rearing densities that we use in production level programs. And so before I jump into the experiments, I'm going to give you a little bit of background of why I think rearing density could be driving this, could be driving the ability for selection to act. So hopefully you remember this graph from earlier. So this is just 8 panels of that performance trade-off figure. So we have the number of returning offspring per parent, and then their per capita offspring success. So remember the parent 70 and the parent 18. This is the exact same panel, except we've broken it out into different years. So this is 95, 96, so on and so forth. Now what I want you to notice is in 95, there isn't a statistically significant trade-off. And you can see that borne out by the line there. It's pretty much a flat line going all the way across. However, in 96, there was a statistically significant trade-off that went through all of the other years as well. And as you can see, this performance trade-off really starts to get strong in 96 and 97. And then by 98, when these fish were being reared at a much higher density, you can see this performance trade-off is there in all of these years. Now, one environment that we were able to correlate the strength of this trade-off with is production class size. So in 95, 5,000 fish were reared. 96, 26,000. 97, 50,000. 98, 60,000 on average. And then I cut off the next 4 years, but they are between 50 and 60,000. So they have that really high rearing density and that really strong performance trade-off. Now, what makes this production class size and rearing density even more compelling and even more suggestive is all of these classes over the years were reared in the same tanks, at the same facility, with the same food, by the same people. You couldn't get more similar, to be honest with you. And truly the only difference was the number of fish in the tank. So here, that performance trade-off might be driven by this increased rearing density. And so from this, I came up with 2 hypotheses. The first, that increased rearing density does cause performance trade-offs in the hatchery environment. And so what we have here on the x-axis is my 2 density levels, and then on the y-axis is a fitness trait. My fitness trait is going to be juvenile body length again, just because we have all this literature showing larger you are at time of release, higher probability of survival. So what I have plotted here is 3 different families. Family 2, Family 1, and Family 3. You can see Family 2 is the biggest in high density, family 3 is the smallest in high density. And then they flip-flop. So what this indicates is that the rank order performance of the families changes from low to high density. So the winners in high density are not the winners in low density. And we assume that low density is a more natural-like condition where selection might be reduced. The second hypothesis I had was that high rearing density increases the opportunity for selection to act. Now, the way we measure opportunity for selection is via this ICC statistic. That's the interclass correlation, and what it is a measure of variation between families compared to total variation. And this is within an individual tank, or within an individual rearing unit. Now, graphically, what this looks like is in high density, we have these 3 families, the blue family, the red family, and the green family. We expect these families to be smaller than the fish at low density just because of the main effect of density. But we also expect large differences between the families. So there's a big white space between blue and then red and green. In low density, we have those exact same families. We know their mean body size is going to be larger, but the important part for this part of the analysis is that the space between the blue and the red and the green is much, much smaller than it is at high density. What this would equate to is in low density, you have a low ICC value. In high density, you would have a high ICC. So at this point, I set off on my next set of experiments, and the important part about this set of experiments is I'm no longer comparing hatchery to wild fish. I'm no longer using 2 different fish types. The rest of these analyses are based on family level differences. So I use the same type of fish for both my experiments that I'm going to be presenting here. And to keep things even simpler, to create families, I just used 2 parents, and that was 1 family. There was no spawning matrix involved. I should also mention that I did all of this work at the Oregon Hatchery Research Center down here on the Alsea River. If you get a chance, definitely go. It's an awesome place. And so I had 2 treatments, high and low density. My low density was housed in large tanks, so these were 12-foot diameter tanks, and my high density was housed in small 6-foot diameter tanks. So unlike the Hood River, where they varied the number of fish per tank, I varied the tank size and not the fish number. And so that's an important distinction that we want to keep in mind going forward. But this is still a test of density, so I continued to trot on. And so just as with the growth rate, I replicated this experiment twice. In 2012, I had a shorter duration experiment of 6 months, and unfortunately I only had 6 families. I started off with 10. We had an issue with one of the rearing flumes in the indoor wet lab that led to some bad juju, and I lost 4 families. So it was a bit smaller than I would have liked, but hey, that's life. In 2013, I reared the fish for a full 12 months, and the reason I did that was to capture the full production cycle that is used by ODFW. So these fish went from egg to that smolt size, that 150 to 200 millimeter size, in that single year. And this year, the fish gods were on my side. I got to use all 10 families, and as you can see, literally 1,000 fish. Big sample size. Just as in the growth rate, I measured their fork length at either 6 months or 12 months, depending on which year it was, and I took a piece of fin tissue to determine their genetic, to determine which family they came from. Same methods as the growth rate, so I used genetic parentage analysis with this Solomon software program. After I sorted all of the juveniles back to which family they came from, I had a few different analyses. To see if the opportunity for selection increased in high density, I simply used a Welch's t-test. So the good old t-test that you learn in Stat 511, the first thing you see. To see if there's a genotype by environment interaction present, which informs us about that performance trade-off, I used another linear mixed effects model. This time, because we're interested in family level differences, my response was average family fork length, not individual fish length. And that would depend on density that you were reared in, the family from which you came from, the interaction between those 2 terms, which is what would inform us about this genotype by environment interaction, and then my random term was tank. So this accounted for correlation within a tank as well as differences between tanks. So jumping right into results, what did we find? Well, here on the x-axis, as usual, density with low and high. and then we have that ICC, so the intraclass correlation on the Y. Each one of these triangles is an individual tank. So in 2012, I had 2 low-density tanks, and I had 2 high-density tanks. In 2013, I got to add a tank, so I had 3 low-density and 3 high-density. Now, although it may appear that these points are higher than these, and those are higher than those, it's not statistically significant. So we found no evidence that high rearing density was increasing the differences between families that would increase the opportunity for selection to hatch. Now, one thing I do want to bring to your attention is in 2012, I only raised the fish for 6 months. And between 15 and 20% of the variation in body length is attributable to simply which family you came from. Now, what was truly shocking, to be honest with you, was in the 2013, when I reared the fish for a full year, 30 to 55% of the variation in juvenile length you could attribute to simply which family you came from, regardless of density. And for the biologists in the room, 30 to 55%, one factor explaining that much variation is simply insane. So looking at the genotype by environment interaction, looking at that performance trade-off, in 2012 we didn't find evidence for a statistically significant effect. In 2013 we did, but I'm going to argue it's not biologically significant. So again, low and high density on the x-axis, and then mean family fork length on the y-axis. So this blue family is a family reared in high density, and then this blue family is the same family, just reared in low density. And as you can see, the lines connecting blue to blue, red to red, so on and so forth, all pretty much have the same slope. So if we were expecting a significant interaction, this blue family would probably be way up here, green might be pushed down a bit, light blue might come up a bit, and because we don't see that, we didn't find an interaction. In 2013, the statistics said there is a significant genotype by environment interaction. But what I want to call your attention to is the rank order performance of these families, of these different colors. So if you're this pinkish coral, that lime green, the purple, or the yellow, you're going to be in the top 4 no matter what. If you're this black and orange, go bees, or the blue, you're going to be in the bottom 3 no matter what. And if you break this out by tank, the same thing happens across every single tank. And it doesn't matter what density you're in. So that pinkish coral was the biggest family in all of my tanks, low density or high density. So what did we get from this? What are the main take-homes here? Well, from the first part, I didn't find that density increased the opportunity for selection to act, but I did find this incredibly large amount of variation in body size at 1 year is due to simply which family you belong to. And so, graphically, we expected these large differences between families in high density, but what we also found was that there are these large differences between the families at low density. And this was another surprise result, just like growth rate not being under selection. And again, we did find a genotype by environment interaction, however, it's weak, and I'm going to argue it's not biologically significant. So at this point, you know, I'm like 4.5 years into my PhD, 5 years into my PhD, and I go, okay, so haven't found evidence for growth rate. Density doesn't seem to be doing what we think it should have been doing. So let's go back to the drawing board. And went to the whiteboard in my office and started thinking about, well, what do we know about these performance trade-offs, about the fitness traits that we know have an effect on survival, and how do my results play in to that story? And so for the last time, I promise you I'm going to make you look at this graph. So I just want to remind you of this performance trade-off, and that body size, the larger you are at least, the higher survival you have. And the logistic shape of this curve is going to be important in the next slide. So I came up with this new idea, this new hypothesis. So here in the top panel, we just have that probability of survival curve. And then here on the bottom panel, this is a completely hypothetical figure. That's why there's no numbers, so don't jump down my throat. So here we have 10 families. So this black dot there is that same family. So the families are the same rank order, just as what we found in my density experiments. So farthest left is farthest left, farthest right is farthest right. The only effect of density is to shift this distribution up 30 millimeters. So everybody gets boosted by 30 millimeters. There's no difference between families. Now, me in particular, I was focused on selection in the hatchery environment itself, but what I hadn't really considered was selection after release and how that might play into this story. So humor me, and let's say selection happens at this point in a particular year. What this means, because of the logistic shape of this curve, pretty much all of the families below this dotted line are going to drop off the map once they go to the ocean. They're going to have really low survival rates, and you're not going to see very many of those fish returning as adults to spawn. So what does this look like after selection happens? In high density, you lose 8 out of the 10 families you reared in the hatchery. They simply don't come back. And this is shown, we have evidence that this effect actually happens. When you have a return of hatchery fish, it's dominated by a very small number of families. In low density, what would happen, I hypothesize, is that you get more of the families above this threshold size for high survival, and therefore when you have these hatchery fish returning, it's not 2 families, it's 8 families. Now what I want to point out here is that you're still going to get the families that do really well in the hatchery here and here. They're going to still come back in really high numbers. But the important part is these 5 families. Those 5 families don't have traits that make them as good a hatchery fish. They don't grow as large in the hatchery. But those fish are likely the traits that, those are likely the fish that have the traits that would make them have higher reproductive success in the wild environment, in the river environment. So, to wrap this all up, a couple of take-home messages here. So that trait under selection portion of my thesis, we didn't find evidence for growth rate under selection. I think it could have been because I didn't stress my fish enough, and it's really hard to replicate that production level dynamic at an experimental level. The second part, looking at the environment driving selection, I found that density didn't influence the ability for selection to act within the hatchery, but I do think there is a potentially large main effect of density on among family selection afterwards. And with that, I'd like to thank you all for coming out, and I'd love to take any questions.
So at this point in the presentation, we take questions from the general audience. The members of the committee will hold their questions until we retire for the private defense, which we'll do back in Worthy Hall. So now, please, any questions you have?
Are you going to be able, do you think it's feasible to test your null hypothesis by releasing fish from really dense and not dense rearing environments?
Yeah, so we have the technology to do it. The issue is it's really expensive. So what Mike and I are actually discussing this now. So we're going to do this novel hypothesis in the next couple of years. And so what I'd like to do is we can insert acoustic tags into fish. And so those have individual ID codes, and then we'll set up receivers throughout the river and throughout the estuary. And so we'll be able to track the smolts as they leave. And then one nice thing with steelhead is there's really low survival through the estuary, so you lose a lot of fish before they even hit the ocean. So instead of waiting 2 years for all these fish to come back, we can get a really quick look to see if there are large survival differences, or look to see if the number of families that make it out to the ocean is higher in low density than it is. Did I say that backwards? It would be higher in low density and lower in high density. Dave?
So I'm thinking about that family level selection model there at the end, which is really interesting. But the one thing I'm trying to wrap my mind around is, isn't it the case that hatchery fish always grow at a faster rate and larger than the wild fish, the completely wild one?
So if you're in the hatchery, you're released. So generally, hatchery smolts are larger than wild smolts. However, I don't personally know of any data that's looked at 2 wild fish with different ancestries. So a wild fish with 2 hatchery parents versus a wild fish with 2 wild parents. I don't know if there's data out there that you can actually look at that at small length.
So maybe that's an unknown, but it would be interesting to know where those 10 families in the wild, with their wild growth rates, would fit within the context of that model.
Absolutely.
Do you think that the effective size on survival is primarily like right after the fish go into the ocean and then they're just getting hit really hard, or do you think that that effect really appears when you're just with it?
So I think it's a combination of things. So the smolt migration is really energetically costly. So a larger fish is going to have more reserves to draw from, and so a lot of people equate the oceans to sort of the bountiful old country buffet, right? Tons of food everywhere. So, but if you get out to the ocean at the wrong time of the year, or you miss the zooplankton bloom or whatever it is, a larger fish is going to have a larger reserve to sort of mill around and figure out where the food is, as opposed to a smaller fish. It's simply going to perish earlier just because it doesn't have that same fat reserve or same energy reserve. That'd be one hypothesis.
Neil, I commend you for being a geneticist that never used the word that I didn't know he was talking about. My actual question, though, is in the first half of your experiments, could you control for how long the fish was in the ocean? It seemed to me that the size of the parent would largely be independent of the dynamics. It would be based on how long they actually spent out in the salt.
Yeah, so the Hood
Is there a
River
way to
fish.
control for that?
So the Hood River fish, the majority of fish, by and large, are 2 salt fish, and you can tell when it's a repeat spawner, whether it's a 3 salt or a 4 salt, just by their body size. And so that 92 centimeter hatchery female could have actually potentially been an extra year in the salt. For the second half, I used Celeste River fish, and all of those fish were generally the same size. So I did not specifically test to see if they were different ages, but I don't think there was a huge amount of variation there.
Is there any literature that would suggest that we've selected for a certain age or time of the fish spending?
Not to my knowledge. So in hatchery Chinook, it's known that if you release a class of hatchery Chinook, a lot of males return after a single year at sea, whereas in the wild, we don't see that. So that is likely influenced by the selection pressure that they experience in the hatchery, but what that is, I'm not sure to tell you.
So there seems to be some evidence that for a given body size athlete, just given small size thoughts on why?
Because the hatchery fish are reared in such a novel environment, so there's no predators, abundant food, and you're reared with 50,000 of your best friends. That's obviously a much different environment than what a wild fish is going to experience. Wild fish, I would argue, is a little bit more hardened, a little bit tougher, street fighter type. I don't have a great explanation for it, other than the different environment that the 2 fish experience as juveniles likely plays a large part in that.
So since Haley didn't ask it, I'm going to ask it. What about trout and resident fish? I mean, do they play a role in this dynamic at all?
So for the hatchery selects for steelhead fishery, and what's really impressive is these hatchery managers who run the shows have figured out how to manipulate growth rates to get their class of Omicus to be steelhead and not residualized. So the one thing I didn't tell all of you for the non-fish people is steelhead can also be steelhead and rainbow trout are the same species, same fish. From a single family, you can get both steelhead and rainbow trout. So it muddies the water substantially. That's why I didn't talk about it. But it could have an effect. And Mark Christie has done a lot of work in this Hood River pedigree, but unless you have one of these really long 20-year thousands of fish pedigree, it's really hard to tease those effects apart. Last question, Larry.
What effect is what you've learned practically on hatchery fish techniques?
So we haven't been able to nail down what's driving fitness fitness loss yet. But once we do actually figure out what is driving these differences between the families, that's when we can then go and say, okay, for these conservation programs, can we change the hatchery environment to alleviate those differences? And at that point, hopefully we're going to have hatchery fish that return and have the same reproductive success in the wild as wild fish. And that's what everybody wants. So that's really where we're going with it. We just need to figure out what's driving the fitness in the hatchery.